Hello, can you guys hear me?
Yes, sir.
Okay, great. Let's get started. Um, Share my screen. You can see my screen as well. Is my screen visible as well?
Yes.
Okay, great. Let's get started. Good evening, everyone. Today we'll be discussing previous year's week six questions. We'll also go over week four and week five questions very briefly since we've missed them. But let's begin with week six. My name is Ashwant, and I'm one of the PMRF TS for this course. Okay, so the first question is microtubule polarity plays an important role in what? Cell migration, cell polarity, cell shape, or both A and B, which is cell migration and cell polarity. What do you guys think? So microtubule has polarity, and that polarity is very essential for certain things. Uh, what are they? Cell migration, cell polarity, cell shape, or both A and B, cell migration and cell polarity. Okay, let's see. So the both A and B. Yeah. So yeah, you're correct. So microtubule polarity plays a very important role in not only trafficking proteins but also it gives cell polarity. So the microtubules are oriented along the cytoskeleton of the cell, and because they have a polarity, the cell ultimately has a polarity, and this polarity helps in trafficking proteins to different regions of the cell, trafficking translated proteins to different regions of the cell, and so on and so forth. But also, it helps in cell migration. So, when you consider neutrophils or macrophages, where they migrate to different parts of the body, microtubule polarity plays a very important role in the direction in which they migrate. Okay. Actin polymerization and branching requires what? ATP only, ATP and ARP two-third, two and three, GTP and ARP two and three, or GTP only. Any idea? So during cytoskeletal formation, actin polymerization requires, does it only require ATP or GTP, or does it require any other protein complex along with ATP and GTP? Uh, Here we are not talking about the spontaneous actin polymerization. We are talking about assisted or uh, mediated actin polymerization and branching. It requires both ATP and ARP23, right? So ARP23 is what results in this 70-degree branching, and so when ARP23 binds, there is this 70-degree nucleation, so formation of a new nuclear center for actin polymer to extend. and that's what results in this branching and elongation, and it requires ATP as well for uh, polymerization. Okay. Lamins are an example of what? A type of microtubule, different structural organization of actin, an intermediate filament, or all of the above. What are lamins? Are they a type of microtubule? Are they a different structural organization of actin itself? Or are they an intermediate filament? Or are they all of the above?
Option C.
Intermediate filament, yeah. So if you look at this schematic representation of cell cytoskeleton, so all these red fiber-like structures that you see are essentially lamins. So they form these intermediate filaments through which the cytoskeleton of the cell progresses and grows. Okay. Which of the following statements is not true? All motor proteins have directionality. Microtubule plus end is pointed towards the plasma membrane. ARP23 nucleates new microtubule filaments, or microtubules are the central support in flagella. Which of the following statements is not true?
Option
Sorry, your voice got cut. Can you repeat that again?
B.
Option B. Microtubule plus end is pointed towards the plasma membrane. You think that's the incorrect option? Okay, so it is in fact true that microtubule placent is pointed towards the plasma membrane, and all motor proteins do have directionality. This is something we've just sort of discussed, which is ARP23 nucleates new microtubule filaments. It sort of nucleates actin filaments, not the microtubule filaments. So microfilaments and actin filaments is what ARP23 nucleates. Okay. Uh, which motor protein drives the bending of cilia and flagella? Dynein, kinesin 1, kinesin 3, myosin. What do you guys think? So cilia and flagella in bacteria have, or prokaryotes have this bending motion. And what drives this bending motion? So dynein is what drives this bending motion. So if we look at the microstructure of the cilian flagella, at the base, what happens if you look at the structure across a cross section? What happens is you see this A tubule and B tubule, right? So these are sort of flanked by these uh, uh, thread-like structures. So these are what are the dynein arms, and essentially what they do is they bend the flagella in one direction. So they turn in one direction, and that results in this bending motion uh, that's observed in both cilia and flagella. So dynein is what drives this bending motion in cilia and flagella. Okay. Which of the following statements about microfilaments is true? Microfilaments are the thickest class of cytoskeletal fibers. Microfilaments are made up of globular actin. Microfilaments break under tension easily. Microfilaments are only found in cells lacking microtubules. What do you guys think? So is it true that microfilaments are only found in cells lacking microtubules? Cells usually contain both microfilaments and microtubules, right? So that's an incorrect option. Microfilaments break under tension easily, which is not true. In fact, they're quite strong. They are basically polymerized actin that has been stacking on top of each other and forming this very robust microfilament. So they do not break very easily under tension. And microfilaments are the thickest class of cytoskeletal fibers. That's also not true. Microtubules are probably the thicker class of cytoskeletal fibers. So essentially, the only correct option is microfilaments are made up of globular actin, which if you look at the structure of microfilament, so there is this G-actin that comes and binds to the microfilament and gets polymerized and forms this uh, fairly relatively thick microfilament, but not the thickest class of cytoskeletal fibers that goes to microtubules. Okay, which of the cytoskeletal components lacks polarity? Intermediate filaments, microtubules, actin filaments, none of the above. What do you guys think? Which cytoskeletal component lacks polarity? So we know microtubules have polarity. Do actin filaments have polarity? Actin filaments do also have polarity and they keep extending in one direction. So both microtubules and actin filaments have polarity. So what does not, the only cytoskeletal component that does not have polarity in this list is intermediate filaments. So they do not have any polarity. Intermediate filaments like the lamins we previously discussed. So they do not have a polarity. Okay. The homologue of tubulin is found in bacteria is what? MERP, Crayus, or FTSZ? Or is it the case that there are no tubulin homologues found in bacteria? What do you guys think?
Option C.
Yeah, so FTSZ is the homologue for tubulin found in bacteria. Okay. The role of motor proteins in cells is to activate cytoskeleton in the cell, move cargo vesicles in the cell, initiate microtubule and actin polymerization, or maintain stability of the microtubules. What do you guys think is the role of motor proteins in the cell? So it's in the name itself, motor proteins. Uh, what do you guys think? Do they help in activating cytoskeleton of the cell? Do they move cargo vesicles in the cell? Do they initiate microtubule and actin polymerization, or do they maintain the stability of microtubules?
Option C.
Initiate microtubule and actin polymerization. Okay. Not really. So the main function of motor proteins is to move cargo vesicles in the cell. So they carry things from one polarity of the cell to the other polarity. So if you might have seen this very famous YouTube video. Let me see if I can pull it up. Just give me 1 minute. Yeah. So essentially, what they do is they walk along these microtubules carrying huge cargoes within the cell. So this here that you see kinesin, that's a motor protein. So it keeps carrying this huge cargo on its head, and it keeps walking on the microtubule from one end of the cell to the other end. So essentially, that's what motor proteins are. Okay. Which of the following drugs inhibit microtubule polymerization? Nocodazole, vincristine, vinblastine, or all of the above? What do you guys think?
E.
Sorry? All of these drugs, in fact, inhibit microtubule polymerization to varying extents. So, if we look at the mechanism of action of nocodazole, what it does is it results in microtubule disruption and it inhibits microtubule formation. So that's what nocodazole does, and vincristine and vinblastine are together called vinca alkaloids. And essentially, what they do is there is this state of equilibrium or equilibrium between microtubule and free tubulin dimers. And what happens is when vinca alkaloids, either vincristine or vinblastine, are present, whenever there is free tubulin dimer, they go and bind to this free tubulin dimer, and this results in this vinca bound tubulin dimers, which sort of form this paracrystalline aggregates, and they do not form this microtubule. So that way they prevent the microtubule polymerization. So all of these drugs prevent microtubule polymerization. Okay. Do you guys have any questions? We can briefly discuss, very briefly go through the previous week's questions as well. If you guys have any questions, uh, bring them up. We can, yeah, Ajay has a question, I think. Okay, so people have been typing in the chat. I have not been noticing. Sorry. Let me see if I can just share the PowerPoint presentation week so that I'll be able to see the screen as well. Okay. Yeah, Ajay, go ahead. If you have any question, please go ahead.
Sir, I am continuously writing in the chat box.
Yeah, yeah. I'm just seeing. So the sound was not playing, so I didn't know you people were writing in the chat box. Okay, so if thalidomide is one option, is it also inhibitor? I do not know what thalidomide is. Let me check. Sold under. Okay. Mechanism.
Thalidomide was used for the morning sickness in early pregnancy.
Correct, correct. So there was some controversy with the.
So, so that started with the patients developed like hypoplastic hands, short hands,
Correct, correct,
condition
correct.
of
So
copomelia.
yeah, so I guess there are two racemic mixtures
Means
of this
according
drug.
to the thalidomide use, means it was used early, was causing more severe damage. If it was used late, then it was causing less damage.
No, so
But
I think
later on, it was used as an anti-cancer drug. That's why. So I thought it has the same action like vincristine, vinblastine.
right, right, right. So at least as far as I know, what happened was there were two racemic or two enantiomers of this thalidomide, and if you use one, it was fine, but if you use the other one, it caused all these various deformations in the body and so on and so forth. So yeah, yeah, I guess if that's given as an option, you can take that as well. Yeah, good point. Okay. Uh, okay. So let's continue.
What is the difference between actin that is present in skeletal muscles and smooth muscles and other cells? Do actin present in the other cells also? That's what. And what is the difference when it is present in the
Correct. Correct. So
skeletal muscle, smooth muscles, and what action it perform? The
yeah,
cytoskeleton is just laying down and Along with the myosin, it always have that contraction. Means
Correct, correct.
shortening of the that's
Yeah.
maybe that contraction may be of two types, isometric or something like that. Measurement remains same. It decreases.
Sorry, I cannot hear you properly. Uh, so you're asking what is the difference between actin that is present in smooth and skeletal muscles, right? Hello, hello.
Sir, actin filament
Huh.
present in smooth muscle, skeletal muscle, and other cells.
Correct, correct.
Because
So,
only
eh,
I know about skeletal muscle. Along with myosin, it
mhm.
helps in contraction. Means the size of muscle increases or decreases. Same thing happens with the cell
Yeah,
or
yeah.
not. That
So same thing happens with the cell, but you have to keep in mind that cell also has these focal adhesions and so on and so forth. That so actin is present in cells as well. So essentially, if you look at the actin that's present in smooth muscle, skeletal muscle, or the cell, it's all the same. And the way so the main difference comes from the fact that the way the actin and the microfilaments and the microtubules are organized, right? So even in smooth muscles and skeletal muscles, actin and microfilaments and microtubules are present, and they are organized in such a way that it's a tissue level phenomenon. But in cells, what happens is the amount of actin that's present in normal cells, the amount of actin that's present is relatively low, and it's mostly used to maintain the cytoskeleton of the cell. Again, this varies from cell to cell. So some cells might require frequent changes of their uh, cellular boundaries and structures. So, for example, if it's a macrophage, it requires this constant change in the cell shape. So, the amount of actin and first of all, the amount of actin would be less, and there is active microfilament remodulation that happens, and focal adhesions are less. But for some other cell that need not change its shape so much, if I were to, so let's say something along the lines of an osteoclast or an osteoblast. So there, more or less, the cytoskeleton is fixed. So there is no, there's only maintenance of actin that happens. Does that make sense?
yes, sir.
Yeah.
And second, my question is, if fimbria, philia, cilia, these are same terms or these are different terms? They are used interchangeably.
Actually, no. So they are different. So there is a huge difference between cilia and flagella, for instance. So let me pull up that image. Let me see. Where is it? Yeah. So usually bacteria have. Okay, I have to share the screen. Yeah, so usually bacteria have one flagella generally. They might have two or three, depending on the species, but flagella are essentially these elongate structures. So if you look at them from the sense of function and from the sense of function, they are very different. But biochemically, they would be more or less similar. So cilia is essentially this very short hair-like structures all over the bacteria, whereas flagella is a tail-like structure towards one end of the bacteria. It can have multiple flagella as well, but generally most bacteria have one to two flagella. So that's the difference between flagella and cilia, and functionally they are very different. So flagella has this propeller-like motion, so it can rotate and it can do this uh, bends and waves so that it propels the bacteria forward. Whereas cilia is more how do you say this? So it's more of a propeller motion. So if you see, it will start sort of vibrating and rotating, and that's what causes this motion. Is that whereas flagellate can sort of uh, rotate across its axis, longitudinal axis, and as well as it can do this wave-like motion to propel the bacteria forward. Yeah, so there are a lot of functional differences, but structurally or biochemically, essentially, all of them are same. Does that make sense?
So just a little about cilias. They are present in the lung border also at the epithelium layer.
Correct,
Also, they
correct,
are present in the intestine also.
correct.
So we call, I think at intestine we call them microvilli. Okay.
Microvilli.
Those
Yeah,
present in the those present in the lungs, they are only for the motion, but those present in the intestine, they are also help in the absorption.
correct.
So do their protein composition is same or it is different, because both they are coming from the epithelial surface.
Yeah, so in the intestines, they mostly help with that absorption just by the fact that there is a large surface area that's being given because of the cilia being present, right? So if you have cilia present in the intestine, the surface area is bound to increase, and any nutrients might get trapped, might move slowly, and that will result in more and more absorption. And there is also a lot of bacteria that can grow along with this cilia, beneficial bacteria that can help in absorption of several minerals and vitamins. So essentially, the key difference is that it provides a very vast surface area for absorption to happen in intestine. In lungs also, they do help in oxygen, a lot of oxygen to get into blood. But yeah, not so much. There's not a lot of absorption that happens with the lungs, right? So it's not absorption is not an important criteria in lungs just because there is no nutrition or anything that goes into the lungs. It's just air. So it provides an ample surface area for the air to diffuse through the lungs and into the blood. But in intestines, it helps in absorption because there is a lot of nutrition that's present on one side and uh, your cells on the other side, along with bacteria that can use this increased surface area to grow. Biochemically, I'm not sure if there's a huge difference. I can check. I'm sure there are some differences people have reported, but yeah.
Do flagella of bacteria also differ? Means in one bacteria's flagella is different from other bacteria's flagella.
Chemically or organize or when it comes to the organization, not so much. So these are very conserved proteins. They are very essential for the bacteria to survive because if a bacteria needs to have flagella, it needs to move and source its food, and that's how it's going to survive, right? So they are very highly conserved proteins. So you would not expect much difference between the organizational structure of the uh, of these motor additions, but uh, mutations might exist and there might be slight differences. Yeah, so I cannot see if I'm trying to find if there's any difference between the cilia in lungs and intestine, but right now I cannot. Does not seem like people have reported much on it. But yeah, so essentially both of them are the same, but just because they're present in different environments, they serve different functions. Yeah. So the size of the flagella might vary from bacterial species to species. So some bacteria might have very large flagella, some bacteria might have very short flagella, and so on and so forth. Some will have one, some will.
Where bacteria require flagella, where they want to move, because just multiplication is enough from cell to cell.
Right, but so you have to understand that bacteria for the most part live in a famine environment, right? So they do not very easily come across food. And depending on the species of the bacteria, or not just bacteria, any prokaryote for that matter, the environment of the prokaryote, they might have to move to source food. So it's not like food is present all around them. There are bacteria or prokaryotes present in volcanoes, salt water, oceans, everywhere, right? So some of them need to scavenge and get food.
If this bacteria want to go with this flagella, how much distance they are going to cover?
For the bacteria, it might be so. It's not like they're going to cover kilometers and kilometers, but so see, you have to understand the world of prokaryotes is a feast or famine world. So there is either plenty of food in the surrounding environment, or there is no food at all. And if there is food, the bacteria might have to move by a few micrometers to get the food, especially if the bacteria is present in liquid environments, such as fluid environments, such as rivers, oceans, so on and so forth, right? So it has to sort of move to get the food. So it might not move meters for kilometers, but for micrometer movements, the flagella is good enough. And I think there is a very well studied example also. So if you look at the uh, feeding of amoeba, uh, which is a prokaryote, so
I'm
it
going to cite because pseudopodia basically they prepare for eating the food, not for the movement.
correct,
Though
correct.
it is called pseudopodia, but it is for the eating movement. No? So rather than flagella, it is just a sense organ rather than locomotor organ for the bacteria.
Sorry, you're saying flagella is a
Sense
sensory?
organ, like sensing something.
Not really. So its major function is movement. Why the bacteria has to move? I do not know the exact reasons, but I at this point I'm just conjecturing. So it might be that it has to go to a point to get some food, or I don't to move to a favorable environment. So let's say there's a bacteria in river. So eventually the river will go into ocean. The bacteria might not be able to survive in the ocean, so it has to have a movement mechanism to sort of keep it from going into the ocean. So that might be one reason to have a flagella, so on and so forth. And please, example of E. coli and typhoid. Okay. uh,
Or typhoid, both I think contains flagella or flagellar antigen.
Correct. So typhoid has flagella. It might, in fact, play a very huge role in the infection of the establishment of infection of for the bacteria as well, right? So if it's going to enter your bloodstream, it might have to go somewhere, or it might have to evade something. So then flagella comes into picture and helps the bacteria move quickly from point A to point B.
Discussion, I think it's not for going from one place to another, rather than clinging to one place. Like typhoid bacteria want to cling at the intestine and they
Correct. So
multiply
so
there.
yeah,
The
that's all.
opposite is true.
Yeah, so that's also beneficial. Flagella can help with clinging to one place as well. So, yeah, that's also true.
The first question I was not knowing. I think I joined late.
Let
Can you
me
please
see.
repeat? Or
Uh, so, microtubule polarity plays an important role in what? Is it cell migration, cell polarity, cell shape, or both A and B?
thank
So,
you, sir.
yeah, so
Answer
essentially,
you itself, though, technically.
both cell migration as well as cell polarity. So microtubules might play a role in cell shape, but here we're talking about the polarity of the microtubule, which will uh, give the cell its polarity and also help in cell migration.
The cell migration is intracellular or intercellular.
In not intercellular, so cell migration as in cell moving from point A to point B. So it needs to have the cell needs to have a sense of direction, right? So if the cell has to move from point A to point B, it needs to have a sense of direction in which it's going. So there the microtubule polarity comes into picture and will help in the movement of the cell. And when it comes to cell
So,
polarity, there
sorry to
is,
ask, maybe a foolish question, but what it achieves by cell polarity?
yeah, so
Microtubule?
correct. So cell polarity is very essential for the intracellular movement that you're talking about. So let's say I'm a cell, I just translated this very new protein. I want it to go to cell membrane. Now, how can I ensure that? So there are several strategies. So there are several redundancies. So I can uh, post-translationally modify the protein, and I can also ensure that the protein goes to the cell membrane by looking at the cell polarity. So we were just discussing about, yeah, I think it's right here. So there are these, oh, you can see. Anyway, so there are these kinesin proteins that carry cargo from one pole of the cell to the other pole of the cell, right? As they walk on the microtubules. So I can ensure that this protein goes to the cell membrane through the cell polarity. So it becomes very vital for the cell to maintain cell polarity to move intracellular cargo from one point to another point with proteins like kinesin and so on and so forth. Motor proteins are what they call it.
Uh, Thanks, sir.
Yeah.
Just wanted to know the difference between plant cell and animal cell at the cellular level. I think cell biology is for both plant and animal cell.
Okay. So,
What's the basic difference apart from the cell wall?
right,
Because the plant can grow the branches, human cannot grow the fingers.
right. So humans cannot
What is
grow.
that thing stem? Because
Yeah,
if suppose stem cell is present in my finger, I will grow my finger if it is cut. That's not
correct,
possible in human beings or any other organism.
correct,
But it is
correct.
possible in some wall lizards. If the tail is cut, they will grow the tail.
Yeah, great question. So, I don't know if they've covered this in the class, but when you talk about stem cells, there are several different categories of stem cells. So, if you look at a stem cell that has just been produced and that's totipotent meaning it can form the entire organism. So this particular stem cell can form the entire organism. So its DNA is very naive. So there are no epigenetic markers to suppress a particular set of genes in the stem cell yet. So it is totipotent and it can produce the entire organism. So plants have a lot of totipotent stem cells. So even if you cut some part of the plant, depending on species again, so for instance, for rose plant, if you just do a stem graft, if you cut a stem and put it in the soil, that's enough to generate roots. That's enough to generate the entire plant. So this is because they have a lot of totipotent stem cells. When it comes to humans, humans do not have any totipotent stem cells. They have multipotent stem cells in most cases, where so after totipotent comes pluripotent stem cells, which can generate most of the organism except brain stem and brain tissue and nerve cells and heart cells. So these are pluripotent stem cells, and after that they have so pluripotent stem cells in humans are essentially the post blastocyst post blastocyst stage, where there is a nerve nuclear center that has already formed and a heart nuclear center that has already formed, and the remaining cells, remaining 12 or some cell odd number of cells can generate the entire human being. So those are pluripotent stem cells. What humans mostly have are called multipotent stem cells, which are, for example, hematopoietic stem cells in your bone marrow, which can generate specific lineages of cells. So these can produce blood cells and immune cells constantly, and so on and so forth. So that's essentially the reason why humans are not able to regenerate these organs through stem cells. Although, admittedly, liver is known to sort of have a little bit of this regenerative ability, mostly because again, these are multipotent stem cells that are present in the liver in humans. They have been organized in such a way or have been epigenetically silenced in such a way that they can produce only liver. So it all depends on the epigenetic changes that these stem cells undergo within different organisms. Yeah, so that's essentially why you're not able to grow back your limbs. It's yeah, so that's that. Whereas lizards and all, if you cut the tail, they can generate the tail back because there is a lot of multipotent stem cells there which can generate the tail. Humans do not have so many multipotent stem cells across all the tissues. So if you take out the blood from a human being, he can generate the blood. So essentially, blood, you can think of blood like part of the a percentage of human body, right? So if you're removing that percentage of human body, they are able to regenerate that percentage. So humans do have some regenerative capacity, but
Blood generation is because we need it for lifelong, because RBC
correct.
don't have nucleus, and they die. Average lifespan is around 120 days.
Yeah, so it's not just RBCs, immune cells as well, and so on and so
Okay.
forth. But essentially, the reason we are able to regenerate these cells is, be it RBC,
We
neutrophils, blastocysts, so on and so forth, is because we have these hematopoietic multipotent stem cells that are present in the bone marrow. Yeah,
Sorry to interrupt, but this bone marrow transplantation is used to cure many disease,
right.
especially I think leukemia were first tried. I don't know whether success or not.
Yeah,
Because of
so
this,
bone marrow transplantation is used successful in curing leukemia, HIV infection, and so AIDS, and so on and so forth. But uh, very costly. Yeah. Go on. What's your question?
question. That's why one means since my childhood, I'm wondering about this. Ki a plant can grow another plant. A human being cannot grow another neck. But in mythological stories, if you see some demons, if their hands is cut, immediately it will grow the another hand. If throat is
Correct.
cut, so I was just wondering, ki he's a sort of a plant who can have that, maybe I don't know the exact word, extended morphogenesis you can call it, because our human beings morphogenesis ends in the fetal life. It cannot go beyond that. But I am still have that imagination of my child who's 10 to 12 years old. Like you cut a leaf, another leaf grows. Like you cut a throat, another neck will come. In
Correct.
mythological was all possible. So
Yeah. So
that's
so
why
so
this question I am.
yeah.
Sorry if
No, 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 no. It's fair enough. In imagination, everything is possible, right? So, I can write a story where I can myself regenerate and I can write a book on it. But we do not know how verifiable that information is. What we know is the amount of or the different lineages of stem cells that humans have. And humans have relatively fewer amount of stem cells. And remember, all these are evolutionary trade-offs, right? So the idea is that we've all come from a same branch at some point, and we've diverged. And we have made this evolutionary trade-off to have less stem cells in an effort to have something else. Now, what that something else, we are not yet 100% completely sure. We think it's most probably a lot of brain mass and brain tissue and so on and so forth, and the maintenance of the body architecture. Uh, and since we do not really require a limb to have a good enough function in our society, we do not necessarily. So it's not like if we lose a limb, we are useless. But for a lizard, if it loses a tail, it might not reproduce. That's one of the key disadvantages that the lizard might have because it has been evolutionarily select. The tail has been evolutionarily selected to not only so if you look at the function of the lizard tail, it will help in not only just the balance of the lizard, but it also helps in making sure that the lizard survives. So it can run and it can hide away from predators. It can attract females based on the size of its tail and so on and so forth. So tail is a very important part of the lizard. Whereas for humans, limbs might not be a very huge, might not have a very huge role because we live in a society, and in our society, even if one person, one individual does not have a limb, it's not like the end of the world for him. He can still reproduce, and evolution only selects for reproduction, right? So, yeah, essentially, we have made some trade-offs to lose this totipotency and pluripotency and so on and so forth. But yeah, and the reason plants are able to regenerate is because they have this totipotent stem cells present within them. And that's essential for them because, in fact, if you look at the way plants reproduce, being able to regenerate the entire organism from part of their organism plays a very important role in plant reproduction, especially if a plant is not able to fertilize and so on and so forth. So, yeah.
So
And
just another related question is, there are stem cell banks are there because some representative has come when my child was about to born. They will collect the stem cell, keep it, and when something mishap happens, it can regrow any organ.
mhm.
So please, can you elaborate how many stem cell banks we have in India? Just an idea, means it's not. I'm not asking exact how much it cost.
Correct, correct. So I do not. You cannot regenerate every part of the human body from stem cells. It's still not scientifically possible. In fact, there is a lot of so very recently, one of the very famous papers where they have generated this pluripotent stem cells from fibroblasts has been proven wrong. So it's still uh, one of the golden questions in biology. Can we reverse? So essentially, stem cells become normal cells through multi-lineage divisions and what do you say, epigenetic modifications, right? So they lose their stem cellness and become more and more specialized. So if even my skin cell at some point is a stem cell, it has undergone multiple rounds of division, and throughout the process of division there are epigenetic modifications that keep happening to it to generate this skin cell, and in the process it has lost the stemness. And some people have the idea of so we can in fact reverse this process. So we can in fact reverse these epigenetic modifications and produce stem cells back from skin fibroblasts even. But we are still not able to produce a pluripotent or a totipotent stem cell from any of the cells in human body. So that's one. And the other thing is you will not be able to generate every part of the human body from a stem cell. In fact, it's very hard to do so. I think most of the stem cell banks are scam, at least for now. Things might change in 100 or 200 years from now. But at present, there's no point in storing your stem cells unless you would want to use it for yourself in the future if you have some leukemia or some of these cancers where you will have to replace a majority of your bone marrow and so on and so forth. But yeah, I do not know what the cost of storing stem cells is and what the cost of how many stem cell banks there are in India though. So stem cell storage cost. They're saying 23,919 plus 51. Okay, so it's relatively cheap, I guess. 21 years cost to store stem cells for 21 years, it costs around 50,000, and for 75 years, it costs around 75,000. And every year, post that, I guess there is every year 23,000 or 24,000 renewal cost.
I think yearly charges are 50,000, sir.
Is it? Okay. Yeah, so it says 21 year storage span at 23,990 plus 34,000 for 34,000. I don't know what that means. I guess if you decide to store for 21 years, you will have to yearly pay 23,990 plus a base fees of 34,000 or something like that. I don't. Anyways, yeah, so for the most part, they are scams. It is very rare that you will use the stem cells. In some cases, they have been very useful, especially when you do not have a proper competent person from whom you can borrow these stem cells. But yeah, I do not know how beneficial they are currently. Maybe 200 years in the future, they might prove to be very beneficial. Any
Thanks.
yeah, no problem. Okay. So I can so we'll I guess we'll go through week five questions very briefly, and I'll upload all these. So I'll upload week five and week four as well on YouTube as well as Google Sheets, so you guys can go through the slides. But yeah, so let's jump into it. So the first question reads: fluidity of a cell membrane decreases with what? Is it increase in glycolipid content in the membrane, or decrease in unsaturated fatty acids in the membrane, or decrease in saturated fatty acids in the membrane, or increase in saturated fatty acids and unsaturated fatty acids in the membrane? So, what does membrane fluidity depend on?
So not able to see the screen of question.
Oh, sorry. I think I said the wrong. Are you able to see now? Yeah. So, what does fluidity of a cell membrane depend on? Is it the glycolipid content or the saturated unsaturated fatty acid content? So, the fluidity of a membrane mostly depends on the saturated unsaturated fatty acid content of the membrane. So, uh, essentially what happens is if a membrane contains a lot of unsaturated hydrocarbon tails, the packing is inefficient, so there's a lot of membrane gaps in fluidity as the membrane can move around, uh, and the membrane becomes more fluid. But if it's saturated fatty acids, what happens is the packing is almost perfect, so very closely packed. So the membrane then becomes viscous. So this is one of the major functions of cholesterol in your body, where cholesterol maintains membrane fluidity of all your cells. So yeah, so decrease in unsaturated fatty acids results in decrease in membrane fluidity. Okay. Uh, Plant cell wall is composed of what? Peptidoglycans and cellulose, cellulose only, chitin and hemicellulose, cellulose, hemicellulose, and pectin. What do you guys think? Yeah, so cellulose, hemicellulose, and pectin. Essentially, that's what the so if you look at the structure of the plant cell wall, you can see this pectin and cellulose, and there's hemicellulose as well. Okay. Which of the following statements is true? Gram-positive bacteria have only cell membrane. Gram-negative bacteria have an outer membrane surrounding the thin peptidoglycan layer. Gram-positive bacteria have an outer membrane surrounding the thin peptidoglycan layer. Or gram stain is retained by gram-negative bacteria.
A.
Okay. Okay. So not true really. So both gram-positive and gram-negative bacteria have an outer peptidoglycan layer. So if you look at the cell membrane, there is a peptidoglycan layer that surrounds this cell membrane both in gram-positive and gram-negative bacteria. But what gram-negative bacteria additionally have is an outer membrane, so a lipopolysaccharide membrane. Essentially, that's what results in the gram stain. So if you stain with a gram stain, what happens is the gram-positive bacteria they take up the gram stain along with the gram-negative bacteria, right? So, uh, but when you do the washing, what happens is the gram-positive bacteria retain the gram stain, but the gram-negative bacteria get rid of the gram stain. And when you do crystal violet staining, counter staining, That's when the gram-negative bacteria get uh, counter-stained with crystal violet. Yeah, so essentially gram-negative bacteria have both of them have an outer membrane apart from the cell membrane that they have, which is a peptidoglycan membrane. But beyond that, so the peptidoglycan membrane is very thick in gram-positive bacteria, but the peptidoglycan membrane is thin in gram-negative bacteria, but they have an outer lipopolysaccharide membrane that surrounds this peptidoglycan membrane. Okay. Secondary cell wall is present in which of the following organisms? Plant cells, animal cells, fungi, or viruses?
Plant cell.
Yeah, so plant cells have a secondary cell wall. Okay. Uh, Microtubules are made up of what? We know microfilaments are made up of actin. What are micro?
Alpha and beta.
Yeah, alpha and beta tubulin are what make up these microtubules. Okay, so what is the function of vacuoles present in plant cells? They support and protect the cell. They control what enters and leaves the cell. They control the cell volume, or they store water and mineral ions.
I think B.
So you're saying they control what enters and leaves the cell? Okay, so actually, these vacuoles' major purpose is to store water and mineral ions for the plant. So they harbor a vast amount of water and mineral ions for the plant so that it does not dehydrate and run out of nutrition. Okay. Uh, okay. MS Dhoni, Rishabh Pant, and Sanju Samson all playing in one team is an example of dash that is also seen in cells. I don't watch cricket, so I have no idea who these people are except MS Dhoni. But
Redundancy. Second. B.
oh, okay, great. Yeah. Okay, so dynamic instability is an inherent property of what? Microtubules, actin, both A and B, none of the above. What do you guys think?
That is both A and B.
Okay. Okay, so actually dynamic instability is an inherent property of the microtubules, not so much actin. Even though actin depolymerization occurs, it occurs only when there needs to be cell movement or significant cytoskeletal remodeling within the cell. So, but microtubules have this dynamic instability that's inherent within them. Okay, which of the following cells lack a cytoskeleton? Prokaryotes, eukaryotes, both A and B, none of the above. Do eukaryotes have a cytoskeleton and do prokaryotes have a cytoskeleton?
None of the above.
Yeah, so both essentially both of them have a cytoskeleton, so none of the above. Okay, which of the following is a function of cytoskeleton? Maintenance of the shape of the cell, positioning of the organelles in the cell, both A and B, none of the above.
Both A and B.
Right. Yeah. So they help in both maintaining the shape of the cell as well as positioning of the organs within the cell. Okay. Yeah. I guess that's all the questions there were for week 5. If you guys have any questions, let me know. But we can briefly go through week 4 as well since we've missed that one as well. Okay. So we'll go over the week 4 questions. The first question reads, which of the following statements about the endosymbiont theory is correct?
Screen, sir, please.
Oh, again, sorry. Week 4. Yeah. So, which of the following statements about the endosymbiont theory is correct? Mitochondria evolved from photosynthetic bacteria while chloroplasts were from aerobic bacteria? Or mitochondria evolved from aerobic bacteria while chloroplasts evolved from photosynthetic bacteria? Both chloroplast and mitochondria evolved from anaerobic bacteria, or both
C.
chloroplast and mitochondria evolved from photosynthetic bacteria. Okay, so you're saying both chloroplast and mitochondria evolved from anaerobic bacteria. Uh, untrue.
B, sir.
Yeah, yeah. So essentially, mitochondria have this ability to produce or generate NADH and NADPH from oxygen through the electron transport chain, right? So they have to be aerobic, and chloroplasts need to have this photosynthetic ability. So yeah, okay. Which of the following is not a difference between a prokaryote and a eukaryote? That is, which of the following is shared by both? Is it the size, organization of DNA, using ATP as a form of energy, or mitochondria? What is shared between prokaryotes and eukaryotes? So definitely not the size.
C.
Yeah. So using ATP as a form of energy is common between prokaryotes and eukaryotes. Okay. Dash is the ability of the microscope to distinguish between two adjacent objects as distinct and separate. So if a microscope were to able to
Resolving
distinguish,
power.
yeah, so that's the resolving power of the microscope. Okay. What is the ability of a molecule to absorb light of one wavelength and emit light of the same or longer wavelength called? Is it chemiluminescence?
Fluorescence.
Yeah, fluorescence. Okay, so which of the following cell types contains a plasma membrane? A plant cell, animal cell, bacterial cell, or all of the above?
All of the above.
Yeah, all these cells contain a plasma membrane. Okay. Typical size of a prokaryotic cell is in the range of 100 to 200 micrometers, 1 to 10 micrometers, zero point 0.1 to zero point 0.5 nanometers, or 10 to 100 micrometers.
Memory based, it's difficult. 1 to 10, I think.
Yeah, yeah. So 1 to 10 micrometers is what the typical size of a prokaryotic bacterial cell is. Okay. Which of the following statement is true? Eukaryotic cells contain DNA, whereas prokaryotic cells lack DNA. Both prokaryotic and eukaryotic cells contain nucleus bound by a nuclear membrane. Prokaryotic cells lack membrane bound organelles, but eukaryotic cells possess membrane bound organelles. Prokaryotes have a cell wall made up of chitin, and eukaryotes have a cell wall made up of peptidoglycans.
C. Third.
Yeah, yeah. So prokaryotes, by definition, do not have membrane-bound organelles, whereas eukaryotes have them. Uh, yeah. So what organism has its cell wall made up of chitin? Do you guys know?
Fungi.
Yeah. So fungus usually contains cell wall made up of chitin. Okay. Which of the statements on organization of DNA in prokaryotes and eukaryotes is false? In eukaryotes, the genomic DNA is packed into chromosomes. Prokaryotes carry a single double-stranded DNA. In eukaryotic cells, the genomic DNA is condensed and held in a region called nucleoli. In prokaryotic cells, the genomic DNA is not separated from the rest of the cell by a nuclear membrane. So,
B.
which of, sorry?
B. Prokaryotes carry single double-stranded DNA.
Okay. So, do eukaryotes have their genomic DNA condensed and held in a region called nucleoid?
Is not present.
Yeah, so prokaryotes do have a double-stranded DNA, right?
Prokaryotes have double-stranded DNA. In nuclear DNA is not present.
Correct. So they do not have nucleus. Instead, they have something called a nucleoid. Whereas eukaryotes have nucleus, they do not have a nucleoid, right? So if I show you, let me see. Yeah. Uh, yeah. So essentially, a prokaryotic bacterial cell has this region where the condensed nucleus is present. It's called a nucleoid. Whereas uh, eukaryotes have nucleus, so a very well defined membrane like structure. Yeah, I guess this is a much better example. Yeah, so this spatial region where the genetic material is present in a condensed form in prokaryotes is called the nucleoid, but eukaryotes have an actual nucleus where it's membrane bound and several other differences.
Sir, there is a dark portion inside the nucleus of eukaryotic cell. What do we call it?
This part, nucleolus, I did not understand the question. So you're saying,
Okay, so I confuse it with nucleus. That is nucleolus. Sorry.
yeah, yeah. So nucleolus is essentially the very condensed or non-expressing DNA wherever it's present within the eukaryotic nucleus. Okay. Yeah, I guess we've covered more or less. Oh, sorry, one more or two more. Let me see. Yeah, so in eukaryotic cells, the genomic DNA is condensed and held in a region called nucleoid is false. Okay. Which of the following features supports the idea of mitochondrion being an endosymbiont? Mitochondria is a double membrane bound organ. Mitochondria carries its own DNA. There are examples of similar codons coding for different
All
amino
of the above.
acids. Yeah. So essentially, all of this observations led to the justification of the endosymbiont theory. Okay. What are the What are the two key differences between plant and animal cells? Presence of cell wall and chloroplast in plant cells, presence of vacuoles and ribosomes in animal cells, presence of chloroplasts and mitochondria in plant cells, presence of vacuoles and mitochondria in animal cells. What do you guys think? Yeah, so essentially cell wall and chloroplasts are present in plant cells, whereas ribosomes are present in animal cells as well as plant cells, and are present in plant cells. Chloroplasts present in plant cells, but mitochondria is present in both animals and plant cells. And once again, mitochondria is present in animal cells, but are not present in animal cells. So the two key differences are plant cells exclusively containing cell wall and chloroplasts. Okay, I guess we have covered all the questions then. Do you guys have any other questions in mind? Okay, if not, then I guess we can end the session here. I hope I was able to answer at least some of the questions that Ajay had, but yeah, see you guys next week. Take care. Bye.